Hello and salam sejahtera. We are here to brief on the human replication helicase, the CMG complex, as the target for anti-cancer therapy. It is the main point of our discussion for DNA replication and the cell cycle assignment. Let's take a look at the first slide. The picture shown proteins that involve in DNA replication, which is the main subject of our presentation. And to be specific, take a look at CMG complex that consists of MCM27, GINS, and CDC45. We all are very familiar with the function of helicases, which unwind the DNA duplex during DNA replication. Through some research, scientists found out that helicases of pathogenic organisms can be a therapeutic target to treat infectious diseases, and as for human DNA helicases, it can be a potential target for anti-cancer therapy. Here, we can see a familiar figure of DNA replication process that we all used to learn. But have we ever go deeper into finding out the structure of helicases? Now, why don't we go and take a look at the detail of the helicase, or to be exact, the CMG complex. What is CMG complex? It is a multi-subunit protein that functions as replicative helicase. This complex made up of the assembly of two subcomplexes and one protein. The protein is heterohexameric MCM27, while the subcomplexes is CDC45 and GINS. Assembly of all three subcomplexes and protein form a heterotetrameric complex, which is the CMG complex. Since it is a human replicative helicase, it is a potential target for cancer therapy. This is the process where MCM27 form a double hexamer in a shape of closed ring, then activated by CDC45 to initiate DNA translocation mechanism and unwind it. Why helicase as the potential target? It is due to the fact that helicases work together and involve in a variety of processes related to DNA such as replication, repair, recombination, and even telomere maintenance. Any failure of DNA duplication and repair leads to loss of genome integrity, which causes cancer and other diseases. This is the image of gene profiling that proves the tumor growth causes by the expression of MCM. What should we do with helicases in order to treat diseases? In this study, scientists ought to find the inhibitor of helicases to control the replication of viral DNA. This method is the alternative way to treat immunocompromised patients suffering from genital herpes that caused by herpes simplex virus or HSV. A few compounds have been identified as the inhibitors of helicase primase complex and also applied for human clinical trials. For now, antibacterial drugs that target for DNA helicase is relatively novel. This is one of the examples of diseases that can be controlled from any further development through its helicase primase level itself. Studies on the mechanism of DNA replication had been extensively carried out using purified protein of Escherichia coli. Based on it, pathogenic strains of E. coli can be treated using inhibitors of DNAB. Similar ideas applied to human DNA helicases in order to restrict the growth of cancer. Olaparib is an anti-cancer therapy that has been approved by FDA as monotherapy for ovarian cancer. Here's the replication process in prokaryotes with DNAB as its helicase. In eukaryote, replication process is more complex with a loading mechanism. Now, these slides explain about RECQ helicases family that involve in genome integrity maintenance. Any genetic defects in these helicases are linked to disease prone to develop cancer. A small molecule helicase activity inhibitors that displayed anti-proliferative activity had been identified, which can help in inhibiting RECQ helicase. This image shows that RACQ superfamily are all helicase, which prevent through gene mutation and helicase assay. Replication stress is any obstacles that delay, prevent, or terminate the DNA replication. Increasing the replication stress in cancer cell can help in its treatment. Here are the emerging approaches that will collapse the DNA replication forks, causes the cell death during mitosis, and impairment on the regulation of the DNA replication.
Diagram shall explain the interference with ATR-CHK1, VWEE1 kinases, and the inhibition of histone deacetylases in pancreatic cancer.
CMG complex catalyze the unwinding of DNA in eukaryote. Expression levels of MCM down-regulated in normal somatic cells enhance expression of MCM proteins reported in many cancer cells. The MCM27 complex consists of 6 different MCM ATPase subunits and is activated when it forms a macromolecular complex with CDC45 and the genes complex, which is we call the CMG complex. This is the schematic structure of the CMG helicase from two models, which are the budding yeast from cryo-EM and the Drosophila CMG. Budding yeast show the views from the NTD of the MCM27 and Drosophila show views from the CMG. The MCM25 gate is open as indicated in the MCM27, while the gate is closed by bound to the CDC45 and the genes in the CMG model. CDC45 genes binding to the MCM27 complex surface bridge MCM2 and 5, and the discontinuity between MCM2 and 5 is sealed by a nucleotide which is omitted in the structure for clarity. DNA replication, initiation, and identification of replicative helicase CMG. The CMG helicase complex consists of 11 polypeptides. Takes place on DNA in order to unwind DNA during the replication. Around 30,000 to 50,000 replicators, which we now call replication origin, are activated at each cell division in humans. During late mitosis and G1 phase, the heterohexamer origin recognition complex (ORC) binds to the origin and recruits CDC6 and CDT1 proteins. The resulting complex then loads two MCM27 complexes on two protein origin DNA complex, forming the pre-replication complex pre-RC. CDK phosphorylation leads assembly of the complex onto the chromosome, initiate replication, the pre-initiation complex pre-IC. CMG helicase complex from CDC45, the MCM27, and the genes and MCM10 protein activates the CMG helicase to create replication forks. Two CMGs from double hexamerate MCM27 move in opposite directions with polymerase. Then two CMGs from double hexamerate MCM27 move in opposite directions with polymerase. 6 subunits readily arranged in the hexamerate MCM27 complex, forming a central channel that DNA can access. The human MCM complexes isolated from HeLa cells. The DNA helicase activity was associated with MCM complex containing only MCM47. All MCM27 proteins are essential for the elongation of chromosome replication, indicating MCM27 is the in vivo replicative helicase.
A three-dimensional view of the CMG complex shall be described in this animation video.
The CMG complex, core of the replication protein complex, revealed through the cryo-EM and X-ray crystallography. MCM27 is a torus-shaped by a regular arrangement 6 monomers. All 6 protomers display the same domain organization, rotary translocation mechanism of hexamerid DNA helicase. The N-terminal domain NTD divided into several subdomains. Helical domain A involved in the decay-mediated conformational switch and the activation MCN helicase in archaea. A zinc finger domain provides an interface for the MCM27 double hexamer formation on the chromatin. And the eukaryotic MCN have N-terminal extensions, NTEs, crucial in the decay-dependent regulation of replication initiation. CTD domain of MCN adopts a wing helix domain, WH domain, which is flexibly tethered to the triple plus domain. The WH domain in the archaic MCM control ATPase activity of MCMs allosterically. Cryo-EM analysis reveals spiral cackled ring structure containing a gap between MCM2 and MCM5. The role of SCTD1 acts as a brace that maintains the open spiral structure of MCM27 until ATP hydrolysis.
Through cryo-EM and X-ray crystallography, these above structures are visible. As you can see, the MCM2 to 7 CDK4 are clearly visible.
The unwinding mechanism of the CMG helicase. According to the strand extrusion or side channel extrusion model, the CMG translocate encircling double-stranded DNA and the unwound strand is extruded via a side channel formed between NTD and the triple plus domain of the MCM27. In the steric exclusion model, the CMG encircles only the leading strand template while the lagging strand template is excluded from the interior of the complex. The inside channel of the MCM27 in the CMG is shown to be large enough to accommodate double-stranded DNA. The CMG stalled when the leading strand had obstruction but did not when obstruction were in the lagging strand, suggesting that the lagging strand is excluded from the interior of the CMG. CDC45 acts as a guardian of the MCM2-MCM5 gate, which captures the leading strand in a 3' to 5' orientation. This is the strand extrusion or the steric exclusion models. Model A show the gate aligned and the model show the gate offset. The constitution of the CMG activation and DNA replication reaction. The formation of active CMG in eukaryotes depends on the formation of the two MCM27 on an origin and the conversion of the double hexamerid MCM27 into two single CMG. Through the single molecule analysis using fluorescent tag proteins, sequential loading of two hexamerid MCM27s on DNA was monitored.
The first MCM27 has been recruited via the ORC or CDC6 and the second MCM27 has been recruited with the first MCM27. The study recently published indicates that for loading two MCM27s, two ORC molecules are required. The relative orientation of the ORC binding sites was crucial to be loaded bidirectionally. CMG helicase assembly reaction reconstitution in vitro will screen and test candidate molecules to prevent the formation of CMG. As you can see, this diagram shows the activation of CMG complex helicase as mentioned before. The association between MCM and cancer was first indicated years earlier by finding a rise in the levels of malignant cutaneous keratinocyte tumor in MCM7 positive nuclear cells. This was also confirmed by subsequent research which demonstrated high levels of other MCMs such as MCM2 and MCM5 in different forms of cancer. MCM3 survival has also been detected in various cancers, including salivary gland epithelial tumors and papillary thyroid carcinoma. The association was positive between MCM3 expression level and ovarian cancer malignancy level. Expression levels of MCM7 were also upregulated in a variety of cancer cells. CD45 overexpression is strongly associated in papillary thyroid cancer with tumor sizes and phases. CMG may also be a marker not only for the development of cancer, but also a druggable target for cancer therapy. This table shows the negative correlation between MCM expression levels and its prognostic outcomes. Now let's take a look at the strategies to target the CMG for anti-cancer therapy. In order to decrease the tumor formation, one model is concerned with the downregulation of MCM, as sustainable and elevated MCM's expression will be crucial for quick cell proliferation. Consequently, siRNA suppress the proliferation of cancer cells through the knockdown of MCM components. On the other hand, the other variant of model concentrates on excess MCMs essential for survival of cancer cells, with limited preservation genome stability when the replicative burden is strong. Let's go into the first strategy, which is the downregulation of constituents of the CMG complex. Genistein, a natural non-toxic isoflavone, and trichostatin A, or known as TSA, are found to be the downregulating MC2M expression in prostate cancer as an inhibitor of classical histone deacetylase, or HDAC. Besides, higher protein expression factor levels are necessary for loading and MC27's activation. Treatment of GNK inhibitor restored TSA-mediated decrease of MCM2, which indicates that MCM2 is downregulated by the GNK signaling pathway. The second strategy for targeting the CMG is to directly inhibit enzymatic activity of the CMG complex. Heliquinomycin was originally discovered from the Streptomyces species and was demonstrated to have antibiotic effects on microorganisms and inhibited DNA replication and RNA synthesis in cultured cancer cell lines. Among fluoroquinolones, ciprofloxacin displayed selective inhibition toward yeast MCM27. It has promises in terms of designing drugs only directed at a particular helicase. This table shows the druggable CMG target for cancer and its targeting mechanism. The development of novel strategies targeting the CMG will shed light on the field of cancer therapy. Currently, most strategies are based on the regulation of protein expression and control of the enzymatic activities by small molecules. However, peptides and antibodies could be used as an alternative to small molecule-based therapeutics in downregulating CMG activity. The Depletion strategy has been tested out not only with MCMs, but also with other subunits of CMG. It was proposed that miRNA governs SLD5 expression and was expressed in human bladder cancer due to the reduced level of a candidate miRNA. The inhibition of the CMG helicase activity could take place in a variety of ways. For example, ciprofloxacin is likely to negatively interact with the ATPase active sites of the MCM27 helicase to inhibit its helicase activity via competitive binding. pCNA is similar to the CMG in terms of its role in cell proliferation and acts as a marker for cancer. Prohibiting protein, which suppressed E2F-mediated transcription to inhibit cell proliferation, was shown to physically interact with MCMs and inhibit DNA replication. This figure shows the potentially small molecules or peptides for the CMG targeting sites. The findings that a functioning CMG needs several critical steps. CMG is a difficult target for the production of drugs with a combination of diverse pathways for inhibition. Helicases are emerging targets that have great potential for the treatment of serious human proliferative diseases such as cancers. Identification of novel type drugs targeting CMG, the core of DNA replication and cell proliferation, is of immense importance in developing new therapeutics against cancers. The development of specific assays will provide innovative means to identify small molecules targeting CMG assembly and helicase activity, ultimately contributing for the development of novel and effective cure for human cancers. This is our results of the isolation and purification of CMG complex, which leads to our experiment in report. That sums up the entire presentation. With that, me and my team would like to express our gratitude for your very kind attention. Take care and stay safe.